Hello and welcome to week 9. This week we're going to be learning about the Enterochloride Test and motility. So this week's experiments are all about identifying and characterizing bacteria. So what are some reasons that we would need to be able to identify microorganisms? Well, the first is in the medical field, and this is maybe the most obvious. We need to be able to identify the pathogens that are responsible for infectious diseases. So if you have a patient that appears with an unknown illness, we need to be able to identify that pathogen. The second major reason is in industry. In industry, we might isolate strains of microbes that produce useful products. So, for example, in the biotech industry, there's an enzyme called Taq polymerase that's very commonly used. It's an enzyme that was isolated from a bacterium, and it's used as a reagent in PCR reactions. Now, PCR reactions you might have heard about a lot lately because it's the test that's used to diagnose COVID. But it's a very common enzyme used in biotech because it's used for PCR reactions, and it's an enzyme that was isolated from a bacterium. And then another example are antibiotics that are produced by bacteria and isolated from bacteria. The third reason we need to be able to identify microorganisms is for food production. So we isolate strains for manufacturing and modifying the quality and flavor in foods. And they're used in the production of fermented food and dairy products. So, for example, fermentation is used in the production of alcohol and dairy, yogurt, and bread. And then the fourth reason is taxonomy. So, evolutionary comparisons of how closely related one bacteria is to another. So, making a phylogenetic tree or a family tree to determine how closely related bacteria, different strains of bacteria, are one to another. So why would that matter? Why is that important to do? Well, say you found an important antibiotic in one species of bacteria. Well, a closely related species might have a similar antibiotic, or maybe something even better. Another example why that's important, if you have a pathogen that's related to another pathogen and you don't know how to treat it, you might look at what treatment works for a closely related pathogen. Now, when we do these different tests to identify bacteria, they work because each microbe has their own biochemical fingerprints that we can use for identification. So what does that mean? That means there are enzymatic activities involved in the microbe's metabolism that we can use to identify them. So metabolism, remember, is all of the chemical reactions in a cell or in a body. That being anabolic reactions and catabolic reactions. Remember that anabolic reactions are building up molecules, and catabolic reactions are breaking down molecules. So metabolism, you might think of it only as the breakdown of food, but that's only part of what metabolism is. Metabolism is building up molecules and breaking down molecules. So all those chemical reactions are happening in the cell all the time, and the result of these reactions is that metabolic end products are formed and excreted. Now, we've been talking about these in previous labs, so this is not a new concept to our lab. We use those end products to identify and classify the microbes. So some examples from the previous labs. In the water testing lab, remember that lactose fermentation produced gas and acid, and we saw that as a color change and a gas bubble in the Durham tubes. Those were end products from metabolic reactions that we used to detect what kind of microbe we had. And also we did this in the staph and strep identification lab. One example from that lab, the MSA plates. Mannitol fermentation produces acid, and we saw that as a color change on the plate from red to yellow. So the end products of the metabolism, whether they're acid, whether they're gas, whether there's certain enzymes that change the color of the colonies from one color to another, all those things are results of the metabolism of the microbe that we can use as fingerprints to detect what kind of microbe it is. Now let's talk about the Enterochloride test for identifying microbes. The Enterochloride test is this cool tube that's kind of like 12 agar plates in one tube. So the Enteropluri test is for the identification of Enterobacteriaceae and other gram-negative, oxidase-negative bacteria. And it's this 12-sector tube that allows for 15 biochemical tests. So that means that some of the sections do more than one test. So they might test for a color change and gas production, for example. So each of those sectors has a specific culture media, and to identify the bacteria, you will observe the changes in each sector and note it as either a positive or a negative. And then you use the positivity values that you read from the test to make this 5-digit code that you will then look up in a code book to identify the microbe. And the book will tell you the species of microbe that you have. So can you use this test for the identification of staph or strep? No, because remember that those are gram-positive, and this is a test specifically for identifying Enterobacteriaceae and other gram-negative and oxidase-negative bacteria. This is a list of all the biochemical reactions that are occurring in the Enteropluri test. So don't worry, it's not something you would ever need to know all of them or memorize or anything. They would always be given to you, but I just wanted to show you all the different tests that are being tested in this one tube. So to do this test, you would start out with one Enteropluri tube and a plate of your unknown, and the tube has this long metal wire that goes through it, and you would use the tip of that wire to scrape up some of your bacteria. And then from the other end, you pull the wire through the whole tube so that every sector is getting inoculated with the bacteria. And then you will let that incubate and then record the results. So when you look at your results, you'll look for color changes in each of the segments, and in some of the segments you'll be looking for gas production, and you would use a reference like this to compare what a negative result or a positive result would be. So a negative result would be no change in color, and a positive result would be a change in color or a production of gas. So for example, in the glucose test, in that first segment there, it starts out red. If you see a change in color to yellow, that's a positive test. So you would go through each segment of your tube, and using this chart for reference, you would mark down if you have a positive or a negative result for every one of those biochemical tests. So the glucose test, if it's yellow, that's a positive. The gas test, if there's lifted wax like a bubble underneath, that's positive. If not, that's a negative. The lysine test, if you see a purple result, that's a positive. If it stayed yellow, that would be a negative. And so on. You go through the chart and mark each segment as either positive or negative. Now remember, I said we are going to use that information to determine a 5-digit code that you look up in a code book. So I'm going to walk you through how to do that. You start by dividing the tests into 5 groups of 3. So you can see it labeled here on this chart, groups 1 through 5. And there are 3 tests each. And these are always in order, so you don't need to worry about what tests are in what group. It's always in order given to you. So you check the positive or negative for each test, and you assign a positivity number. So a negative gets a 0. The first test in a group, if it's positive, gets a 4. The second test in a group, if it's positive, gets a 2. And the third test in a group, if it's positive, gets a 1. And I'm going to go over this. We'll do one together so you can see what I mean by that. So once you have those numbers, you add up the group positivity number, and that will give you a 5-digit code. So let's try one of these together. So let's say you did an enterochloride test in the lab with your unknown, that's labeled unknown A, and these are your results. The first thing you do is to divide the tests into the 5 groups of 3. So the first 3 tests, draw a line after it. Next 3 tests, draw a line. Next 3 tests, draw a line, and so on until you have your 5 groups of 3 tests. Then you look at your results, and for each test you go through and check whether it was a positive or negative, and put a check mark in the box positive or negative for each test. Next, you assign each test its positivity number. So remember that all negative tests are 0. So if you want, you can go through all the negative ones first. All the ones that you checked negative, write a 0 in the positivity number. So then go back to the top, and so glucose was positive. So remember, the first test in a group, if it's positive, gets a 4. So glucose gets a 4. Gas is the second test, and it was positive. A second test in a group, if it's positive, gets a 2. And then we move on to lysine. We gave lysine a 0 because it was negative. So then we go on to the next group. Ornithine was positive, and it's the first test in the group. So it gets a 4. The H2S test was negative, so we gave it a 0. Indole test was negative, so it got a 0. Go on to the next group. Adonitol was negative, so we gave it a 0. The lactose test was positive. The lactose test is the second test in that group, so it gets a 2 because it's positive. Arabinose is positive, but it's the third test in that group, so it gets a 1. Moving on to the next group. Sorbitol was negative, so it got a 0. VP was positive. VP is the second test of that group, so it gets a 2. Dulcitol was negative, so it got a 0. Moving on to the third group. PA test was negative, so it got a 0. The urea test was positive. Urea is the second test in that group, so it gets a 2. And citrate is positive. It's the third test in that group, so it gets a 1. Then you simply add up the positivity numbers for each group to get the group positivity number. So for example, the first group is 4 plus 2 plus 0, so that group's positivity number is a 6. So you do that for each group, and that gives you 5 numbers. Those 5 numbers are your 5-digit code. So in this example, we determined the 5-digit code to be 64323. So you will then look up that code in the Enteropluri manual PDF that I will give you. And sometimes there's more than one possibility. So you would write down all the possible identities given for your microbe. Next, let's talk about motility, what it means, and why it's important to test for it. Characterizing bacteria based on their motility is another way to help identify them. So what is motility? Motility is the ability of an organism to move on its own, so self-propelled motion. And being able to do that has some advantages. It can help the organism to acquire nutrients, to move to the best site to colonize within its host, to avoid toxic substances by moving away from them, and also to disperse in the environment during transmission from one host to another. So 3 mechanisms for motility include flagella, axial filaments, and gliding motility, and we'll talk about each of those. Flagella are long whip-like structures that help some microbes to move, and I have a picture here and a list of terms, and I want you to be able to recognize which term goes with which picture. So for all of the terms, they end in trichus. Trichus means flagella. So for A, monotrichus, mono means one, and a monotrichus microbe has one flagellum. And even some human cells are monotrichous. For example, sperm cells are monotrichous. They have one flagellum. So for B, lophotrichus, lophotrichus means they kind of have this ponytail of flagellum on one end. And then C is amphitrichus. Amphitrichus has 2 flagella, one on each end. And then finally D, peritrichus. Peritrichus means it has flagella all around the outer edges. So think perimeter, the outer edge, all along the outer edge. So the direction of the whipping rotation of all these flagella determines the movement of the cell. Axial filaments are these filaments that wrap around the cell and make the cell move in a corkscrew movement. So spirochetes are organisms that can be distinguished from other bacteria by the presence of these axial filaments. And you can see in the picture a diagram of the axial filaments wrapping around part of a spirochete. So some examples of diseases caused by spirochetes are syphilis, which is a sexually transmitted disease, Lyme disease, which is transmitted through ticks, and leptospirosis. You may have heard of, if you have a dog, there's a vaccine for leptospirosis in dogs. And then gliding motility is the ability of certain microbes to move on a surface without any structure for propulsion. What they do is they secrete a layer of slime, and then they can slide across and move along on the layer of slime. So motility is another clue that we can use when we're trying to determine an unknown bacteria. So how can we test to determine if our unknown is motile? Well, one way to determine if a bacterium is capable of motility is to perform a soft auger stab. Now, what that means is that there's auger in a tube, as you can see in the drawing below, and it's softer than normal auger in a plate, so it allows for motility of the bacteria within the tube. And you take some bacteria on a sterile needle and stab it into the soft auger and pull it directly out in a straight line. And once you allow that bacteria to grow, you'll be able to see if that species is motile or not. If it's not, if it's non-motile, you'll see just a line where the bacteria grew and stayed in one place. But motile bacteria would grow throughout the whole tube because they can move throughout the soft auger. Some commonly used motility augers include standard soft nutrient auger. So that's made with less auger than you would use for a plate, so it stays softer. And that can be made with or without TTC. TTC is triphenyl tetrazolium chloride, and that is used to visualize bacterial growth since it produces a red pigment when it's reduced by bacteria. So that can be added to the auger when you make a soft nutrient auger. And then the other two types are SIM and MIU. Those are two other differential media. The important things to know for those are the three things that they test for. So SIM is a differential media that tests for sulfur reduction, indole production, and motility. And MIU augers are differential media that tests for motility, indole production, and urea.